Ribosomes are molecules within cells that facilitate the formation of peptide bonds. They also condense two amino acids into a dipeptide, which form a peptide bond as well. Peptide bonds are where polypeptides are synthesized, which are essentially a chain of amino acids. These amino acids are linked together by condensation. Here I am drawing the structural formula for proline. Proline is an example of an amino acid being genetically modified to create another amino acid, hydroxyproline. The amino acid sequence of polypeptides is coded for by genes. The DNA is stored in the nucleus, yet the polypeptide is produced by the ribosomes. So the messenger RNA is sent from the nucleus to the ribosome, given instructions for how to put the polypeptide together. The genetic code is the sequence of bases on mRNA. This tells the ribosomes which amino acids to use. Because of this, the amino acid sequence determines the three-dimensional conformation of a protein. Every individual has a unique proteome. For example, Rubisco is an enzyme that helps fix CO2, and this is commonly found in plants. Insulin is a hormone that signals cells to absorb glucose and help reduce glucose concentration of the blood. I think the circle I drew is supposed to be a cell, and then the figure I drew is supposed to be glucose. Immunoglobulins are antibodies. I don't know why iMovie messed up the format here, it's too high, but it says immunoglobulins at the top. Rhodopsin is a pigment that absorbs light and is how you see. Collagen are rope-like proteins that give strength. These fibers are found in your skin, blood vessel walls, and parts of your teeth and bones. Spider silk is a protein stronger than steel and Kevlar, and when it is stretched, it expands and is resistant to breaking. Proteins are denatured by a change in pH or heat. This breaks the bonds apart, causing the substrate not to be able to fit back in the enzyme correctly. An enzyme is a globular protein that increases the rate of a chemical reaction by lowering the activation energy threshold, which is basically a biological catalyst. So the enzyme is the weird-looking, almost bean thing I'm drawing, and the substrate is the reactant, and that's going to fit snugly against the enzyme. The activation site is where those two meet. Enzyme catalysis involves molecular motion and the collision of substrates with the active site. Remember back to the video we watched in class of the two kids running around the hallway carrying books, and they crashed into each other, and that's how they started dating. This is similar to the situation that happens with an enzyme and a substrate. This is a normal enzyme, and this is a denatured enzyme. Enzyme activity is affected by temperature. This graph shows that at a certain temperature, the enzymes are denatured and there are more collisions happening at an ideal temperature for the enzyme. This differs depending on the enzyme. Similar to temperature, enzymes also prefer a certain pH. Too much pH or too little, the enzymes will be denatured. The amount of substrates also affect the enzyme activity, because there are only so many enzymes, and not all substrates will be able to collide with the enzyme if there is an abundance. An example of immobilized enzymes being widely used in industry is lactose-free milk. Milk is repeatedly poured over beads with lactase, and because of this, the lactose is broken down into glucose and galactose. This then produces lactose-free milk. Activation energy is the initial input of energy that is required to trigger a chemical reaction. Enzymes speed up the rate at which the reactions can occur. Inhibitors change the active site of an enzyme and prevent substrates from entering. There are two types of inhibitors, competitive and non-competitive. Competitive go right where the substrate should be, and non-competitive go in another part of the enzyme, but still change the active site. End product inhibition prevents a large buildup of product. The allosteric site is where end product binds on the enzyme, which is not the active site. This causes a temporary conformational change, or locking, of the active site. Threonine uses end product inhibition to convert itself into isoleucine.